All organisms interact with other organisms, and some more intimately than others. So I'll be talking this time about parasites and endosymbionts. So this is Genomics Part 6, and you can find the information from this in chapters 30 and 31 in the Integrated Molecular Evolution second edition. Organisms that are said to be free-living or fairly independent, most are not completely independent, but when they can survive on their own for long periods of time, their genomes are maintained because they need all of the functions that are being encoded by those genes. However, when you get a parasitic or commensal relationship, one of the organisms, at least one of the organisms, starts losing some of its genome because it's depending on products from the other organism for some of the things that they need to survive. By the time you get to a mutualistic relationship, both organisms tend to lose sequences, primarily because they're dependent on the other organisms for part of the things that they need to survive. And then when you get to an endosymbiotic relationship, including organelles, it's somewhat different in that the endosymbiont tends to lose genes, and the main organism picks those up. The main genome of the host organism may also lose a few gene sequences that are supplied by the endosymbiont as well. So when genomes are examined, then you can talk about free-living organisms such as E. coli and Haemophilus influenzae. And they have fairly large genomes at 1.8 megabases and 4.7 megabases, and carry around at least 1,500 to sometimes 4,000 and 5,000 genes. If you examine the genome of an obligate parasite, such as Mycobacterium genitalium, it has lost quite a bit of its genome and a lot of its sequences, so it only has 500,000 base pairs of DNA in its genome and carries 470 genes. By the time you get to organelles, they have lost even more of their genomes. Plastids, that is chloroplasts and amyloplasts, etc., which are the same organelle, have been inside of plant cells for about 1.9 billion years, and others have been in about 1 to 1.2 billion years. So they've lost quite a bit of their genomes, having genome sizes of about 120 to 250,000 base pairs, and they carry from 46 to 209 genes. Plant mitochondria, and mitochondria in general, have been inside eukaryotic cells for about 2.4 billion years, and they've lost even more of their DNA. Plant mitochondria are a bit unusual because they carry a lot of non-genic sequences. So they have about 62 genes, but the genome size can range by quite a bit. Whereas the genome sizes of mitochondria in vertebrates, or in animals in general, is usually under 20 kilobases and have about 30 to 50 genes within those mitochondrial genomes. Here are a few other examples. So Bradyrhizobium has 9.1 million base pairs of DNA in the genome and about 8,300 genes. E. coli, about 4.7 megabases, Haemophilus, 1.8 megabases of DNA, and from about 1,700 to 4,300 genes there. Chlamydia, Polynella, Mycoplasma. So chlamydia and Mycoplasma are both obligate parasites, and they have from about 500,000 to 1.5 million base pairs of DNA in their genomes, and from 400 to about, or from 500 to about 1,100 genes. Polynella has a chromatophore. It's like a chloroplast, but it's more recently obtained. So this is an event that took place about 1 billion years ago, versus in plants where the cyanobacterium entered about 1.9 billion years ago. Because of that, it's actually lost less of its DNA. It's down to about 1 million base pairs of DNA and 867 genes. So it has a lot more coding capacity than do chloroplasts. But this is analogous to a chloroplast, but it's a second event that occurred much more recently, about 1 billion years ago. These are events that happened a longer time ago. So for chloroplasts in green algae and plants, they've lost quite a bit of their DNA. They only have from about 130 to 250,000 base pairs in their genomes, and from about 40 to almost 200 genes. Toxoplasma gondii is an alveolate, that is a protist, that has an organelle derived from a red alga. So it's non-photosynthetic, but it was originally a red algal cell with a nucleus, a mitochondrion, and a plastid. It's been reduced even further. It has only 35,000 base pairs in its genome and only 26 genes. And then back to mitochondria, which entered the first eukaryotic cells around 2.2 to 2.4 billion years ago. With the exception of plants, and I'll talk about that in a second, the genome sizes range from about 6 to 80,000 base pairs, and from about 5 to 60 genes. Now, for plants, it's a little different because a lot of the DNA in the mitochondria in plants is non-coding, so they only have about 60 genes. But the genome sizes, even within a family, within the cucurbit family, muskmelon versus cucumber versus some of the other melons, watermelon, they vary quite widely in genome size, although the number of genes is pretty constant at about 60 to 62. So these values can be plotted, that is genome size versus the number of protein encoding genes. And if you do these plots, then the free-living organisms group more or less together, so at the high values. So above about 1,500 genes, and genome sizes are above about 1.2, those are all free-living organisms, or mostly free-living organisms. In the middle range, then you have parasites and endosymbionts, where they range from genomes of about 200 or 300,000 base pairs up to around 1.2 million base pairs. And then they have from 200 to perhaps close to 12 or 1,500 genes. Then you go down even further, and organelles have even less DNA and fewer protein encoding genes. So in general, when you have this evolution from free living to parasites to endosymbionts to organelles, the trend is for reduction in the genome size and reduction in the total number of protein encoding genes. These are some other organisms plotted, and again, you can see the same trend where larger genomes with larger numbers of protein encoding genes, those organisms are free living. Of course, they're interacting with other organisms as well, but they're more or less free living. And then in the middle range, you have parasites and endosymbionts, where you have below 1,000 genes usually, and below about 1.2 megabases of DNA. And then going down into the organelles, where the amount of DNA and the number of proteins keeps decreasing more and more. And in particular, you can see the plant mitochondrial DNA here and the plant mitochondrial DNA there. They both have the same number of genes, around 62. But the genome size varies widely. So even having a smaller genome or a smaller number of genes doesn't necessarily mean that the genome is all that small. Most of the extra DNA, however, is non-coding DNA. This is a diagram from the book showing all of the known and suspected endosymbiotic events. that have occurred that led to organelle development in the past several billion years of evolution. The ones that most people know about and learn about are, of course, the alpha proteobacterium joining with a protist cell that became the mitochondrion, and then the other one where a cyanobacterium was taken up to become the plastids or chloroplasts. There was a second event that we know of now between a cyanobacterium and a protist cell that became the chromatophores in Pollinella. Also, it's suspected that the nucleus came from a planktomycete or something like that, and planktomycetes have nucleus-like structures in them, so that may be the source of the nucleus. And then going back a little further to get a gram-negative bacterium, which has two membranes, several different theories posit that this is from two gram-positive bacteria joining together in an endosymbiotic event to become a diderm, or a gram-negative, having two membranes. And for each one of these endosymbiotic events, then you're joining together at least two different genomes. And then the genomes, in an evolutionary sense, are sorting out which genes are necessary, which ones aren't necessary. The ones that aren't necessary tend to mutate more often and eventually disappear, and the ones that are necessary, although they mutate, there's selection to keep those existing within those organisms. There are several other endosymbiotic events that involve two eukaryotes, and so in the case of the excavata, you have a mutualistic relationship between a green algal cell and a protist that become the euglenoids and eventually the trypanosome line also, which at one time was, its ancestors were photosynthetic, but they've lost that ability and become parasitic. Another group that gained a green alga are the chlorarachnophyta, and those ones are a protist cell and a green alga, and down here, and those are have actually retained their nucleus in that original green algal cell, as well as a plastid, and then they have the main nucleus and a mitochondrion in those cells. And then there's a whole group over here that have taken up red algae, and those include ciliates, heteroconts, haptophytes, cryptomonads, and then you've got additional endosymbiotic events between eukaryotes and eukaryotes to form a number of different dinoflagellates, as well as apicomplexa. And so these include the organisms in the groups the Rhizaria, Haptophyta, Alveolata, and Stramanopiles. And so those are those protists that have gained photosynthetic capabilities in many cases by taking up a red alga. However, some have subsequently lost those red algal symbionts, or they still have them, but they're no longer photosynthetic. So one of those, for instance, is the toxoplasma that I just talked about a few minutes ago in a previous slide. This graph summarizes some of the major events in evolution, and you can see many of them are endosymbiotic events. But starting on the left-hand side, first you have development of RNAs and then eventually DNAs, so that occurred very early on, as well as photon capture with CO2 fixation, photosynthesis. And then you have these endosymbiotic events, first forming the gram-negative bacteria, and then forming the first eukaryotic cells with a true nucleus that may have come from planktomycetes, followed by acquisition of an alpha proteobacterium that could do oxidative phosphorylation to detoxify the oxygen that had appeared in the atmosphere, and also to make additional ATP. And then next is the endosymbiotic event of a cyanobacterium into a eukaryotic cell to become the plastids. And this happened a second time later in Pollinella to form a chromatophore that's like a chloroplast. And then you have a series of endosymbiotic events between different groups of stromatophiles, of violata, excavata, etc., to take on either green algal cells or red algal cells, or in some case possibly both, to become photosynthetic organisms. Before they were not photosynthetic, so this was an advantage to them to become autotrophs. And then one final event to become multicellular. This may have actually occurred back 2 billion years ago. There's some controversy over that, but certainly over the last 1 billion years, there have been a lot of multicellular organisms that have evolved. And in mixing these genomes in these endosymbiotic events, again, there's a mixture of proteins that are formed from those and other products. So there's a lot of possibilities for incompatibility, but there's a lot of opportunity for improving those cells and allowing them to live in different niches and different environments around the world. However, they do give up something, and that is independence. So once they've become symbionts and then endosymbionts and finally organelles, they're completely dependent on one another, on the host organism as well as the endosymbiont. So they can never become independent again. That's sort of the end of that those two independent organisms. So the first event that appears to have happened is the formation of the diderms, the gram-negative bacteria, and it happened probably 3 billion years or so ago. So you have monoderms that have a single plasma membrane and then a peptidoglycan layer, although there are a few without that. And then gram-negatives have two membranes, an outer membrane and an inner membrane, and they vary in their composition. But just to develop a second membrane is difficult mechanistically to explain. But if you explain it as two monoderms coming together, one becomes the endosymbiont and one is the host, then that's an easier proposition to make, primarily because the number of genes involved to make that second membrane is pretty large, and it's hard to imagine how those could have evolved a second membrane just purely by changing slow evolution of the membrane structures they already have. An analysis a few years ago, a phylogenetic analysis, was performed to look at what the two possible organisms were that produced these gram-negative bacteria. And by looking at certain genes and how the genes must have come together, and then statistically analyzing what the best possibilities were, they came out with an actinobacterium, which is a gram-positive bacterium, joining together with a Clostridia, which is in the Firmicutes, to produce the diderms. And then in that case, then the monoderms become monophyletic in a phylogenetic tree, and the diderms become monophyletic in a phylogenetic tree. So it looked moderately promising. And here is the tree that they produced at that time, and note that it's not a standard tree that is branching, but it branches and then comes out with these rings. So it's called a ring tree with these little circular areas in the middle. So in this case, the paper proposed that an actinobacterium came together with a Clostridium to become these double membrane diderms, gram-negative bacteria. And that leads to all the gram-negative bacteria. And then you have other Firmicutes, bacilli, etc., and archaea branching off in a different direction. There was one problem with that is that the photosystems that are present in each one of these groups really was not consistent with this tree. So in their tree, they would have a group of the actinobacteria and a Firmicutes coming together to make the diderms. One of the problems that I've always seen with this is that cyanobacteria, which has both photosystem 1 and photosystem 2, always comes out closer to gram-positive monoderm bacteria, whereas all of the other diderms group together separate from cyanobacteria, and those groups have, some of them have either the type 1 reaction center, or they have the type 2 reaction center, or photosystem 1 or photosystem 2, but not both. So the question is, how did cyanobacteria wind up with both, while the others wound up with only one of the photosystems? And because of that one characteristic of the photosystems, then joining an actinobacterium together with a clostridium, which is from Firmicutes, that just doesn't make sense unless all of the photosystems evolved independently of one another, and that's hard to reconcile. So here's what I mean. If you look at cyanobacterium up there in green, that's a diderm, and that has reaction center 1 and reaction center 2, essentially photosystem 1 and photosystem 2. And then you have the monoderms there, actinobacteria, chloroflexi, and Firmicutes, and then you have all the other diderms. And the diderms are split between those that have only reaction center 1 here, and the ones that have reaction center 2, and those are split. So how can those be explained? Because photosystems take a lot of different genes, and so it's hard to reconcile that those came together many different times in those different sets of organisms. So I set about trying to reconcile these differences, and this is a phylogram that I produced based on what I think is now happening, or has happened in the past. So at the base, on the lower right, you have an ancestral reaction center. This diverged into reaction center 1 in the Firmicutes, and only in a few groups in the Firmicutes, and over into chloroflexi with reaction center 2. And then the actinobacteria lost all photosynthetic activity if they ever had any. You can have then an endosymbiotic event between a Firmicutes and a chloroflexi, and they bring together both photosystems, and that would produce the cyanobacteria. To get to the other groups, so now you have a diderm that's photosynthetic with both photosystems. To get to the other groups, then you have to hypothesize that one or the other photosystems, or in some case both photosystems, were lost. And this isn't difficult to do. Most of the time in evolution, it's more common to lose a function than to gain a function. And on this phylogram also, the 1, 2, 3, 4, 5, 6, 7, etc., those stand for sequential deletions or additions to particular genes that have been measured by other people. So those have been placed on the phylogram. And also nitrogen fixation capabilities have been put on the phylogram. So those are all then consistent with this phylogram, which is, I guess, looks kind of messy or complex, but that's maybe what has happened. There are two other groups that don't fit into this puzzle, and they have always been a problem. So the DTF group, which is the Deinococcus thermus fusobacterium group, and then the negativacutes, which are and a sister group to them, which have been placed into the Firmicutes, but they also are diderms, and they look like they don't really fit into the Firmicutes. And so these have been put into a different category over there, where I've proposed two other endosymbiotic events that produce those, and then you have a tree that is finally parsimonious with regard to the number of membranes, the nitrogen fixation pathways, as well, and genes, as well as the photosynthetic apparatus pathways and genes. So on a few of the slides I showed just a few minutes ago, where I proposed, and other people have proposed, that diderms were formed by an endosymbiotic event between two monoderms. I think that has to be pluralized. That is, there were multiple endosymbiotic events that formed the diderms, not just a single event. This diagram is one that I produced to follow the photosynthetic apparatuses around in these different clades of that phylogram. So you start with an ancestral reaction center on the left, which is based on a sulfur, iron-sulfur center. That then gets duplicated and then elaborated a little more to become the Heliobacteriaceae in the Firmicutes, and that has reaction center 1. Also, you get some mutations occurring, so now there's a pheophyton reaction center without an iron-sulfur group. And that then duplicates and becomes the reaction center 2 that's found in chloroflexi. And also, you get loss of the reaction center in the actinobacteria up at the top. So the major groups appear to have been formed from a single endosymbiotic event between a member of the chloroflexi group and a member of the Heliobacteriaceae within the Firmicutes. And so now you have both photosystems present, and if you go straight over, this becomes then the cyanobacteria type. The cyanobacteria originally were anoxygenic, but they picked up a manganese center that allowed them to get more energy out of this system and more electrons by splitting water into oxygen electrons and hydrogen ions, protons. To lead to the other major groups within the diderms, then the first thing that happens is you get a loss of both centers, and this leads into the Eleusimicrobia and PVC groups, where they have no photosynthetic members. If only the reaction center 1 is lost, then that leads to the Gematomonodetes. When only a reaction center 2 is lost, that leads to Chlorobi and Acidobacteria. And then going around this line, a loss of reaction center 1 in that group, a final loss in the delta and epsilon proteobacteria, a loss of reaction center 2, so those have no photosynthetic members. And then going down the line, just with the loss of the reaction center 1, are the main groups of the proteobacteria, the alpha, beta, and gamma proteobacteria, which contain reaction center 2. This leaves a few groups out of that endosymbiotic event, and that includes the DTF group, Deinococcus thermus fusobacteria, which no members are photosynthetic, and the Hala anaerobiales and negativicutes, which also are non-photosynthetic. So that entails 2 additional endosymbiotic events that lead to those 2 groups. One between an actinobacterium and a chloroflexi, leading to the DTF group, and one between an actinobacterium and a firmicute, leading to the negativicute clade over there. And then this fully encapsulates the monoderms in one group and the diderms in a second major group. Although diderms were formed in at least, it looks like, 3 different events. So now I want to start talking about genomes where there has been a reduction in the the total amount of DNA as well as the number of genes in those organisms. And the first one is a mycoplasma that's in the Firmicutes. And this is one that was isolated from chimpanzees, I think. Genome size is about 580,000 base pairs, 484 genes, 1 ribosomal RNA, and 33 tRNA. So just from the RNAs and the number of genes, you could probably look at this and decide that this is a parasite of some sort. So it's an obligate symbiont of primates. Species of mycoplasma live on different parts of the body, and probably some are living on you right now. And it has genes for DNA replication, transcription, translation, DNA repair, and cellular transport. But it has very much reduced metabolic capabilities. So here is the genome. It's unusual in some ways in that most of the genes on one strand are located on one half of the genome, and the genes on the other strand are located on the other half of the genome. So this was the first bacterial genome ever completed back in 1993. Here's another diagram of the genome. At that time, you could actually put them in a linear form, and for something this small you can, but for anything larger it becomes impossible. So there are 484 protein coding genes, 518 in total, counting the tRNAs and the ribosomal RNAs. The one ribosomal RNA is here, and then the tRNAs are spread out throughout the genome. Also, the colors on here indicate genes within the same classification. That is, they deal with amino acid synthesis, cell structure, etc. And you can see that they're not really clustered to a great extent. There are some clusters, such as this one and this one, but in general, things of the same color are spread out throughout the genome. So here are some of the functions just in a diagram representing the cell. It does have a glycolysis pathway. It also has some nucleotide biosynthesis and some phospholipid biosynthesis. So it can make its DNA. It can do a certain amount of sugar metabolism, and it can make its membrane. But it does have a lot of transporters. So a lot of the transporters are transporting amino acids and other chemicals that it needs inside. And then it has a way to make ATP as well, as an ATP synthase. So rudimentary, but it gets along just fine by living off of the things that it can obtain from its host. So this is a comparison of the Mycoplasma genitalium genome compared to Haemophilus influenza, which has a small genome, but it's a free-living bacterium. So you can see as far as amino acid biosynthesis, it only has one gene for that. Otherwise, it can't make any other amino acid. It relies on the host for that. Cell envelope is rudimentary. Energy metabolism is rudimentary. And all of them essentially are rudimentary functions in each. And in fact, they have about 35% of the gene capacity that Haemophilus influenza has. So they've lost roughly 65% of their genome through becoming dependent on their host. So here's a pie chart of the gene categories that it contains. So it still maintains some genes for creating a cell envelope, that is the cell membrane and some of the proteins for integration into the membrane. Energy metabolism is rudimentary. It has genes for DNA metabolism, repair, and recombination, as well as translation and transcription, although all of those are somewhat smaller than what you would expect from a free-living organism. They also have about 10% of their genome in transport and binding, and those are those transmembrane proteins, transporters that are bringing things into and taking things out of the cell. So a pretty rudimentary genome in this case. The next bacterium I want to talk about is Buchnera aphidicola. It's a gamma proteobacterium that lives in aphids, hence the name. About a 616 megabase genome plus a 2400 base pair plasmid. It has 544 genes, only one ribosomal RNA gene, and 32 tRNA genes. It's an ancestor of E. coli, which is also a gamma proteobacterium. And it does live in cells in the gut of the aphid. Now, aphids eat plant sap, and plant sap is very poor nutritionally, lacks a lot of a lot of different amino acids. But the bacterium has genes for DNA replication, transcription, and translation, so it makes some of its own proteins, as indicated by the number of genes it has. It has a rudimentary metabolic network, but it's lost most of the protein transport functions. It has retained essential amino acid synthesis pathways, and this is probably one reason it's retained in the host, is that it's providing the host with a lot of amino acids. DNA recombination genes have been lost, as well as other DNA repair functions, so it does have a relatively high mutation rate. Here is a micrograph on the left of those bacteriocyte cells which include the Buchnera aphidicola cells within them. And those, again, are producing a lot of the amino acids for the aphid. There's the aphid feeding on a stem. The stem contains mainly some sugars, mainly water actually, some sugars, and just only a few different amino acids and cofactors. So if you or the aphid were to just try and survive on sap alone, you would eventually die of malnutrition. Here are a couple of the genomes of two different varieties of these, or strains of these, including one with a 416 million or 416,000 base pair genome, and the one that's 640,000 bases of the genome. They have similar genes. They're located in different spots. You can see here, though, that they are concentrated on those chromosomes. Another thing that happens with these parasites and endosymbionts is they tend to lose sigma factors and transcription factors. Since they are growing in a very controlled environment, they don't need to react to as many differences in the environmental conditions. So they tend to lose those sigma factors and transcription factors, and eventually some of them have lost all of them, so they're pretty much acting as if they're in a constant environment all the time, which of course they are. This diagram also shows the chromosome locations of the genes on the yellow strand here, and then on the opposite strand, which has the genes colored in orange. Here's a schematic diagram of Buchnera aphidicola inside the host cell of the aphid. So there's a nucleus on the mitochondrion of the aphid cell. And so here the bacterium is bordered by cell wall and cell membrane, so it can make its own cell wall and its own phospholipids for the cell structure. It also has ion transport capabilities for some ions, especially the ones so it can make its ATP, electron transport for pumping protons out of the cell, which can then be imported back inside the cell to make ATP. It has a rudimentary glycolysis pathway and a pentose phosphate pathway as well. But it does make many of the amino acids, so that's probably its main function in the aphid cell. It also has some nitrogen metabolism capabilities and some transport into and out of the cell. But in total, it lacks several functions, so it would not be able to survive outside of those cells. It can't. It doesn't have enough protein encoding capacity to become a free-living organism. Here are the genomes of three strains of Buchnera aphidicola that grow in different species of aphids. And so this is an attempt to show how some genes are lined up in all three. So you can see this blue gene here is in the same location on the same strand of each one of these. And these are all linearized chromosomes, although they're circular in the organism. But then you have other blocks of genes that are unique to one or more, sometimes a pair, of each one of these different strains. So there is some synteny. Synteny means that the chromosome, the genes on the chromosomes line up perfectly in certain locations. But then there are other places where the synteny is not very strong. In fact, there's a lot of unique regions for some of these strains of Buchnera. This gives you an idea of how these different strains have evolved in different species of aphids. Some of the aphids grow only on wheat, others grow on fleshy dicot species and other types of plants. So you can see that as the aphids have speciated, that these strains of Buchnera, which have always been, in this case, endosymbionts of these aphids, also have diverged as far as their genomes to some extent and specialized for each one of the hosts. This diagram illustrates that. So the genes were traced through the three different strains of Buchnera aphidicola, and each one, remember, is in a different species of aphid, and those diverged some 150 to 200 million years ago. So the last common ancestor had more than 638 genes, but then the common genes in these three that have changed, there are 499 of those. So in some cases, the genes are retained, as here in green, whereas for the same four genes have become pseudogenes in these other two strains. And then in another case here in green, those 7 genes have been retained and they've been completely lost in the other strain. Conversely, here, 8 of the genes have become pseudogenes over here, but are retained by these strains over here. And then another 64 genes have been completely lost over here, but retained in along this line. And then you can move your way up looking at genes that have diverged among all 3 of the strains. Here's one where the 22 genes are retained in these 2 strains, but those 22 genes have become pseudogenes in this strain over here. Another case here where 1 gene has been retained in these 2 strains but lost in this 3rd one. And you can go up looking at all these changes, accounting for all of those 499 genes and their changes. The 638 then also indicates that quite a number of them, at least 140 or so, have not changed at all in all 3, so they've been retained. So this next one, Salsia muelleri, is from the Bacteroidetes group of bacteria. It has a very small genome, 244,000 base pairs. 261 genes, 1 ribosomal operon, and 31 tRNA genes. As with Buchnera, it is an obligate symbiont of several sap-feeding insects, and it produces 10 essential amino acids for those sap-feeding insects. It has genes for replication, transcription, and translation, but a very rudimentary metabolic network. Whereas Buchnera aphidicola had genome sizes around 600,000 nucleotides. Here's Solsia muelleri way down here at 244,000 nucleotides, so it's quite small. Here is the glassy wing sharpshooter, which contains Solsia cells within its gut cells. And again, this is mainly, the Solsia is producing amino acids for this insect. This insect also carries pathogenic bacteria with it, plant pathogenic bacteria. So it's a major agricultural pest that is capable of carrying bacteria that will kill fruit trees, especially. As with Buchnera, the genome of Solsia varies according to which species of sharpshooter the Solsia is inhabiting, and so it can range in the 200 from about 240 to up to around 280 or so kilobases for the genome size. Here's the genome of Solsia muelleri. It's a very small genome, as I said, and it has a reduced number of transcription factors. In fact, it only has one sigma factor remaining. So pretty much transcription of this of the genes on this chromosome are pretty much constant throughout the life cycle of the cell that it inhabits. Also note that the genes are very compacted and concentrated, so you see genes on one strand are in yellow and on the other strand are in orange, and for the most part, almost exclusively, if you have a gene encoded on one strand, then you don't have genes encoded on the other strand. They're pretty much mutually exclusive. So Solsia muelleri has one of the very smallest genomes of any bacterium, and it is an endosymbiont. And you can see that the number of genes and gene categories is quite small. The largest category is in ribosomal proteins, so it still needs to make some proteins. It needs to translate the RNA into proteins. So those are necessary, and you also see that the other large category is in the genes to produce essential amino acids. And so that's primarily the function of this organism, also in providing amino acids for the insect, the sap-feeding insect. But Solsia has help from another endosymbiotic bacterium within the genus Baumania. So Solsia produces the amino acids, and Baumania produces other compounds that are necessary for the life of that sap-feeding insect, the glassy wing sharpshooter. Baumania species are from the gamma proteobacteria. It has a genome size of just under 687,000 base pairs. 617 genes are encoded, and it has 2 ribosomal RNA genes and 21 tRNA genes. These species all are obligate symbionts of several sap-feeding insects, including the glassy wing sharpshooter. And it only produces 2 essential amino acids, but it produces many cofactors, also called vitamins. It has genes for DNA replication, transcription, and translation, so it does make its own proteins, but it has a rudimentary metabolic network, similar to that seen in Solsia. Here is a representation of the genome. Again, the outside 2 rings are genes on one strand versus the other strand. And again, as we've seen previously in all of these, the genes are very compacted together, very concentrated. And if you have genes on one strand, you don't have genes on the corresponding strand on the other side. So they're sort of mutually exclusive. The inner ring there is those green lines. That's where all the tRNAs are. And then the two dark blue lines are where the ribosomal RNA loci are. So all of them are pretty much dispersed throughout the genome, as in almost all bacteria. At the bottom, you can pick out some of the processes that are still represented in the genome of Baumannii. This diagram shows that Solcia and Baumannii are pretty much complementary to one another as far as what they provide to the insect and to one another as well. There's a third one. The HC stands for Hodgkinia cicadicola, which was found primarily in cicadas, but also in other sap-feeding insects. So from this diagram in blue on the left-hand side, you can see that Solcia produces enzymes that are within the biosynthetic pathways for lysine, threonine, etc. And for the most part, the amino acids produced by Solcia are not produced by Baumannii and vice versa, so they complement one another. The same can be seen with Solcia and Hodgkinia in cicadas, where they complement each other as far as the amino acids they're capable of producing. On the other hand, Baumannii is capable of producing a large number of cofactors and vitamins necessary in certain functions of the insect, whereas Solcia produces none of those. If you look at Solcia versus Hodgkinia, there's a more limited number of vitamins and cofactors that can be formed by Hodgkinia, but it has worked in the cicadas, so that you can see a little bit of the evolutionary pathways of each of these as they diverged and joined up with different endosymbiotic bacteria in those insect cells. This is a diagrammatic representation of what's going on in the glassy wing sharpshooter cells. So the glassy wing sharpshooter is eating xylem sap, which contains a few amino acids, organic acids, and sugars. The amino acids are primarily aspartate, asparagine, glutamate, glutamine, malate, and then the sugar is glucose. But Solcia produces a large number of amino acids that aren't supplied in the xylem sap, and so it's essentially feeding the glassy wing sharpshooter cells with those amino acids. It also produces a few cofactors, but not many. Those are primarily supplied by the Baumannii cells. So biotin, heme, riboflavin, etc. are produced by Baumannii, and those are fed to the glassy wing sharpshooter cells. So Baumannii also produces some other necessary compounds up here at the top. So altogether, having all three cells, the glassy wing sharpshooter cell plus the Solcia cell endosymbiont and the Baumannii cell endosymbiont, they produce a system that is nutritionally complete so that those glassy wing sharpshooters can survive just living off of the plant sap. So the last species I want to talk about is Carcinella rudii. It has the smallest known genome of any bacterial endosymbiont or any bacterium at all. And it really verges on being instead an organelle rather than just a bacterial endosymbiont. The Carcinella cells live within the cells of these psyllids, and these are also sap-feeding insects, which also carry some pathogenic plant diseases. So their genome, again, is much smaller than any other bacterial genome, and they do end up producing necessary compounds for the psyllid so that they can actually survive on that nutritionally poor sap. Here is the information on Carcinella rudii. It is a gamma proteobacterium, a genome size of about 160,000 nucleotides, which is about like a sum of the bacteriophages. 182 genes with 3 ribosomal RNA genes and 28 tRNA genes. And again, it's the smallest known bacterial genome and is, sort of overlaps with organelles. It's an obligate parasite of sap-feeding psyllids. It is not known how this organism contributes to the survival of the host, but probably some of it has to do with feeding the host amino acids, at least some. Has a high coating density, one of the highest, with a very low GC content. In a previous presentation, I showed a graph where as the genomes of these symbionts and eventually organelles become smaller and smaller, the GC content goes down from a free-living organism having around a 50% GC down to a much, much lower GC content. The genes are shorter than for other organisms, so not only is the genome shrunk, but some of the genes are shorter as well. It has a rudimentary set of genes for replication, transcription, and translation. Many of the ribosomal protein genes are missing, so this occurs with organelles as well. Their ribosomes are actually smaller than those of the bacteria, and they've lost some of the ribosomal protein. So this is more characteristic of an organelle than of a endosymbiont, at least an early endosymbiont. Most of the missing genes may have moved to the host chromosome, and then this is also characteristic of an organelle. So it's sort of on the border between an endosymbiotic bacterium and an organelle. And it cannot make some amino acids, including histidine. Here is a pie chart for the gene categories for Carcinella rudii. A large section of the genome goes towards translation and tRNA synthesis. So it does make its own proteins. It does have some rudimentary pathways. But the genes for DNA replication, transcription are very much smaller. And remember, this has a small genome, so those percentages also indicate a very small number of genes doing those. Other gene products may be coming from the nucleus of the host cell, which is more organelle-like. It is able to make some amino acids, and so that may be its primary function in these cells. And then a very rudimentary set of genes for energy metabolism, probably at a bare minimum just to keep that organelle or organism, whichever you want to call it, surviving within those cells of the psyllids. So that's the end of this section on parasites and endosymbionts, and then this will lead to the next presentation, which is on organelles. And so you've seen already Carcinella rudii looks like it's sort of in that transition zone between an endosymbiotic bacterium and becoming an organelle. And then we'll talk about the major organelles, including mitochondria, plastids, and other organelles. Alrighty, thank you, and see you in the next presentation.